continue those solving questions from the cell structure. All right. In the last previous class, I solved a few, and here is some of these questions. Okay. So okay. Now see this question number 33. Okay. Hello. Question number 33, that select the correct matching in the following pairs. This is a matching, this is with the structure and its function. Okay. One side here, option 1, the smooth ER is synthesis of lipid, rough ER synthesis of glycogen, rough ER oxidation of fatty acid, smooth ER oxidation of phospholipid. Which one is correct matching?
Number 1.
Yes, the number 1, that smooth ER is with the synthesis of lipid. Those cells engage in fat synthesis. Okay, cells which are engaged in lipid synthesis, this have more of smooth ER. Okay, those which are with engaged in lipid synthesis, they have more smooth ER, and those which are active in protein synthesis, say cell engaged in protein synthesis, they have the rough ER more. Rough ER they have more. Okay. Right. So these things, and also within the cells, say this is cell, and in this place, if it is this area is active in protein synthesis, so in this area rough endoplasmic reticulum will be more, and if say in this area it is lipid is synthesized, here the smooth ER will be more. So this one is the synthesis of glycogen. These are actually the carbohydrate synthesis that is not with the endoplasmic reticulum. Okay. Next question. This was a question that was asked in CBSE IPMT 2015. Next is that the nuclear envelope is a derivative of membrane of Golgi complex, microtubules, rough endoplasmic reticulum, smooth endoplasmic reticulum. Okay. Nuclear membrane is derivative of what? Means from where the nuclear membrane is produced. Nuclear membrane is derivative of? Say, if you see the nuclear membrane, So this is the nuclear membrane, and nuclear envelope, we will say. So if this is the nucleus, and this one is, this is nuclear envelope, okay, nuclear membrane, and these are the nuclear pore. This is nuclear pore, right? And here is a nucleoplasm. That nucleoplasm contained the nuclear material. Nuclear material is mainly that chromatin network, and there is also that nucleolus. Okay, this nuclear membrane is continuous with the endoplasmic reticulum. It is continuous with endoplasmic reticulum. So from here, this way, this nucleus is continuous with this till that the cell membrane, the membrane endoplasmic reticulum, it is continuous. In one side, it touch the cell membrane, the other side, it is touching the nuclear membrane. So it continuous, and on the endoplasmic reticulum, we can see this beaded structure. On this endoplasmic reticulum is the ribosome. Okay, this ribosome. So this one, endoplasmic reticulum with the ribosome, these are actually the rough endoplasmic reticulum, and it is continuous. And we can see that on the surface of the nuclear membrane also some ribosomes are attached. Nuclear membrane is double layered. Okay, it is double layered on the membrane which is on the outer side, that is towards the cytoplasm. That side, the nuclear membrane also contain the ribosome. So from here, it is says that the nuclear membrane is the derivative of rough endoplasmic reticulum. Okay, this one is a derivative of rough endoplasmic reticulum, not the smooth endoplasmic reticulum. So next question, let us see this question. You see, this is also came in 2014 CBSE IPMT. The osmotic expansion of cell kept in water is chiefly regulated by mitochondria, vacuoles, plastid, or ribosome. Which one?
Vacuoles.
osmotic expansion of cell kept in water. When the cell is kept in pure water, then what will happen? There will be endoosmosis. Say if this is the pure water and the cell is kept here, the cell concentration, cell cytoplasm is more concentrated than water. So there will be endoosmosis, osmotic entry of water. And this water is moved where? This water move into the vacuole. So this osmotic expansion of the cell kept in water is regulated, chiefly regulated by the vacuole. So vacuole contain, actually in the vacuole there is a more amount of maximum amount of water is there, and in that water there are some dissolved substances, some minerals are there, some enzymes, hormones, etc. are there. So this one, this maintain the osmotic expansion of the cell. So here in plant cell, and these things help in, as there is osmotic expansion takes place, this help initially help in the cell growth, that is the increase in volume. During the earliest stage of growth, these things help a lot for expansion of the cell size or increase of the cell size. Next question here, important function or important site for the formation of glycoprotein and glycolipid. What is glycolipid and glycoprotein? Hello, anyone?
Golgi apparatus.
Yes. No, that is okay. This is Golgi apparatus is the answer. Ah, that's true, but it is what glycoprotein and glycolipid. What is that? Glycolipid and glycoprotein. These things actually, glyco is sugar, okay? Sugar, or it is carbohydrate. And then there is protein plus protein. So protein attached with some portion of sugar, that is the glycoprotein, and protein with glycolipid is lipid plus sugar, sugar plus lipid. So that is these things. Say actually the protein is synthesized on the surface of endoplasmic reticulum, and after that these proteins are processed when it is moving through the endoplasmic reticulum tubes. Okay. Finally, it come to the Golgi complex, Golgi apparatus, and there all the modification, maturation, further processing takes place. Here, the protein, if some of the molecule of carbohydrate is to be added to the protein, okay, that is the complex proteins are formed. So this is this attachment with one molecule, one group of molecule to other group of molecule. All these things takes place within the Golgi apparatus. So function of Golgi apparatus is to for the formation of glycolipid and glycoprotein and many other enzyme protein concentration, increasing the concentration, and their further modification, all these things. And then it is finally packed in a small vesicles. This may be called the spherosome. Okay, those things they are packed, and then finally it is sent to maybe within the cell or outside the cell. So this is Golgi complex is, that is, processing and packaging industry, okay, of the cell. Processing and packaging industry of cell. Then which of these is not the function of Golgi apparatus? Which one? Material packaging, that I
Option D,
told
sir.
this packaging is option B,
D.
D. Yeah, site of protein synthesis. This is not protein, is synthesized on the surface of endoplasmic reticulum, and this ribosome, where the ribosome is present. Okay, so say this material packaging takes place, secretion also takes place. Secretion means those I told the secretory um, uh, vesicles or spherosomes, they are formed, and then finally it is sent out of the cell. And membrane transformation also takes place. Actually, this Golgi complex is a dynamic structure. Whatever is present in the Golgi complex, that is not permanent. The different membrane, etc., that enter into the Golgi complex, and again it come out. So this is a dynamic structure. So the site of protein synthesis is not the function of Golgi apparatus. Next is, which one of the following cell organelle is enclosed by single membrane? Most of the cell organelles, they are covered with a double layer of membrane. Just now I told that the nucleus is bounded by two layer of membrane, is it? Two layer of membrane. The cell also is bounded by, if this is the cell, then in the cell is also there are two layer of membrane. Okay, so all these are double membrane. Chloroplast. This chloroplast has two layer of membrane. Then if we see this mitochondria, mitochondria also has a double layer of membrane. Outer layer is smooth and inner layer forms the cristae. Is it? So there are two layer of membrane. Golgi complex also, that membranes are there. They are also double layer. Nucleus also double layer. So this here, this chloroplast is double layered. Nuclei, they are double layered membrane. Mitochondria, double layered membrane. The lysosome is a single layer bounded organelles. It has only one layer of membrane. Okay. Another one, along with the lysosome, another one is the vacuoles. Vacuole also is bounded by only one membrane, and that one is called the tonoplast. Okay. What it is called? Tonoplast, the membrane of vacuole. So this is a vacuole lysosome. They are single layer of membrane. Beside these, some other structures are also there, like these small vesicles, spherosomes. They are single layer. So you have to remember the double layer, single layer, and those which are not having any membrane, those which are not having any membrane, they are ribosome. Okay, ribosome is without membrane. Ribosome and then centriole. They don't have, they are not having any membrane. The centrosomes, they have amorphous material around the centriole main substances. Next one is with the this is based on the diagram, and here the diagram of mitochondria has been given, and in this you have to tell this labeling A, B, C, D, E. What are those? Go through the sectional view of a mitochondrion showing the different parts and identify the structure A to E. So this structure we have to identify, and the options given here. Say options are given here. A is outer membrane, option A, outer membrane, inner membrane, matrix, D, intermembrane space, C, E is Christie.
Option A, sir.
Option A, sure. A is outer membrane. B, this one, yes. This one is inner
Inner
membrane.
membrane, sir.
Actually, if it was drawn like this way, na, that this one is outer, this one is inner. So this is the inner membrane, and then the outer membrane, and C is the matrix, matrix, the mitochondrial matrix. Okay, D is intermembrane space. Is a D is not showing that intermembrane space. No, D is here pointing the inner membrane. D is this is inner membrane. A is outer membrane. E is the Christie. Then what are the other options are there? Is there anywhere D is showing matrix? D intermembrane space, outer membrane, inner membrane, intermembrane space, D matrix, no. E Christie, E is Christie, outer membrane, B inner membrane, C matrix, D is Christie, no. E is intermembrane space, D matrix, C Christie. Actually, C should be the C is matrix. That is true. If D is, of course, this one, D is mentioning this one. This is not the intermembrane space. Intermembrane space is this portion, see, in between the outer and inner membrane. This is outer, this is inner, and D should be this space between these two membrane is the intermembrane space. Intermembrane space. This is also known as perimitochondrial space. Perimitochondrial space. Okay. It means if this one is coming correct, means the labeling is little bit defect is there. A outer membrane, B. If B is perimitochondrial space, inner membrane, B inner membrane, inner membrane. Then D is intermembrane space. But this labeling is not correct. Okay. Here the intermembrane space will be that in between the outer and inner membrane. That should be, this should be limited to here, not touching the inner mitochondrial membrane. Okay. So this will be the answer here because the others are not totally matching. D is matrix. D cannot be the matrix. Matrix is C. D intermembrane space. This one has a little doubtful. This drawing may be in the labeling that the arrow that is showing, this arrow is not correct. Okay. So you have to answer the most correct one. The others are not totally matching, so you have to answer this one. But here D should be. Say when it is mitochondria is drawn like this way, that is two-dimensional structure. This is the outer, inner, and this is perimitochondrial space. This one. Okay, this is Christie, and this is matrix. Okay. Next is one. The Christie in mitochondria is important because. Question number 72. The Christie in mitochondria is important because.
Increases surface area.
Yes, so this increase the surface area. A lot of membrane is packed within a small space, and this lot of membrane is required for their that the ATPase molecule to be arranged within this, because inner mitochondrial membrane contain the electron transport chain, electron transport system, and ATPase molecule, that fundamental molecules or elementary molecules. So these are the site where ATP is synthesized, is it? So as many membrane will be there, as much membrane will be there, the more this ATPase molecule can be accommodated there. So this one, that Christie of mitochondria increase the surface area of the membrane. Next, why the concentration of number of ions and other material is higher in vacuole than those in cytoplasm? Concentration of ions.
Option A, sir.
Option A.
Yes, sir.
Chronoplast has a number of active transport system uh, that pump ion into vacuole from the cytoplasm. If say active transport, what is does it send substances against the concentration gradient that takes place by active transport system. So against concentration gradient within the cell, that vacuole is there, so here actually in general it should be higher in the cytoplasm, but some of the ions particularly they are present in the vacuole higher concentration due to presence of active transport system. Through osmosis, it will not go through osmosis, water will move here. If water will move here, then this will be diluted. Cytoplasmic ion enter the vacuole through osmotic flow of water. Vacuole is always engaged in the hydrolysis of salt into their ion. Which of the following statement is incorrect? Mitochondria, unless specifically stained, are not easily visible under the microscope. This is true. Actually, mitochondria are very small structure within the cytoplasm. Okay, they are thread-like or rod-like or sometimes granular structure. Hmm. So if to observe mitochondria within the cytoplasm require a special staining, coloring, that usually perhaps genus green is used. So this is true. Physiological activity of cell determine the number of mitochondria per cell. This is also true. A cell will have less mitochondria or more mitochondria, that depend on its metabolic condition, the physiological activity. If the physiological activity is very high and those activity require energy, ATP, If ATP requirement is within the cell or that activity is very high, means they will require ATP, and ATP is synthesized by the activity of mitochondria. So more activity, more ATP required means more mitochondria will be there. Those cells very less active, they have very less number of mitochondria. Again, in case of if we compare the plant cell and animal cell, then in compared to plant cell, animal cell is usually having more mitochondria. In plant cell has less number of mitochondria because in the plant cell ATP production is also carried out by the chloroplast. Is it? Chloroplast, we know in the light reaction of photosynthesis that ATP is synthesized. So the function of ATP synthesis is also carried out by the chloroplast in plant cell, and that's why plant cells have less number of mitochondria in compared to animal cell. The mitochondria divide by fission. Mitochondria it divides. Okay, the outer membrane forms a number of infolding called cristae. This one is the wrong statement. Okay, this statement is true. The mitochondria divide by fission means one mitochondria may be divided into two or three or more mitochondria. Okay, so this is by fission, breaking down into smaller pieces. And this one is wrong because not the outer membrane, it is the inner membrane create the cristae. So answer is this one. Next, next is also say some hello. From some of you that some noise is coming nearby from nearby. Some children are talking, shouting. Please tell them to stop. Hmm. Okay, here come to this question, question number 82. Question number 82, identify from A to F in this. This is the diagram of chloroplast. Okay, so you have to identify this structure. What are these? This is outer membrane. This is inner membrane. These are, this is grana, is it? Then grana thylakoids, and this interconnecting one is the stroma thylakoids, and this is the stroma. This F is F and B both are showing a stroma only. B is granum, no. See here, A is showing inner membrane. Yes, A is inner membrane. B is granum. No. So this one is not. Say you need not to compare all these. Okay, if you know the structure, they are labeling by seeing any one of these that given A, B, C, D, E, whether this is correct or not. Say in B, B is not the granum. Okay. In the option B, what is there? Outer A, outer membrane. No. B, inner membrane. C, granum. C is granum is there. D, thylakoids. Okay. E, stroma lamellae. F is stroma. F is stroma. E is stroma lamellae. This is okay, but this A is outer membrane, B inner membrane. This labeling is not proper. Maybe this line has been drawn, no? These are wrong. C option, what it says, A is thylakoid. No. So this C option cannot be. D, A is outer membrane, B is stroma. C inner membrane. No, this is also wrong. So here in this question, actually in the drawing there is a defect. Okay, this will be up to here. A, B will be up to here. Okay, that is the inner membrane. Outer membrane, inner membrane. E is a stroma lamellae. F is a stroma. C is the granum. D is thylakoid. Right? Then it will be B is the option. B option is correct. Question number 81. Hello? Near someone, I think it is disturbing. Some of the children are playing or 81. Choose the correct pair. Stroma lamellae. Interconnect the grana. This is true. Thylakoid. Singular unit of grana. Thylakoid membrane. Okay. A stroma of chloroplast contain a small double strand. This DS is double stranded circular DNA. This is also true. Ribosome of chloroplast 80S. This one is not correct. Say ribosome of chloroplast is 70S type. It is always prokaryotic type. It is less than 80S. Those chloroplast and mitochondria, they contain ribosome, though they are the organelles of the eukaryotic cell, but this organelle ribosomes are 70S type, not 80S type. So this is wrong.
Excuse me, sir.
Yes,
What does S stand for?
S is sedimentation coefficient. Sedimentation. This one you can see as a sedimentation coefficient. And this is also known as Wedberg's Works unit. Okay. So for that also you can say S. All right. Wedberg was the scientist who actually this S value determined. He has started these things. Okay, this one is sedimentation coefficient. It is called that sedimentation coefficient is that how fast a molecule or a organelle or a substance that sediment in a ultracentrifugation. In ultracentrifugation, okay, in ultracentrifugation it rotate more than lakh per minute. Okay, per minute. The solution kept in a centrifuge tube, and then in this centrifuge it is rotated. That is by applying centrifugal force. The substances having different mass, they can be separated from the solution. And particularly when the cell organelles are separated or different molecules having the different molecular mass, they can be separated through this ultracentrifugation. And in the ultracentrifugation, a particular substance or the matter, how fast it sediment, that it settle at the bottom, that indicate the sedimentation coefficient. This is a physical property of a substance like those density, mass, etc. So this is used as a S value for this also indicate the size of the molecule or the organelles also. Okay. So ribosomes of the organelles, ribosomes are they are 70s, not 80s. So our incorrect pair is D. Okay. Any doubt? This actually centrifugation is a very detailed a physical process. Okay, by gravity we can large substances we can separate by gravity. Okay, I think in chemistry you have studied no separation of substances from a mixture. That one of the method is sedimentation, that the substances they settle at the bottom due to the force of gravity, but gravitational force is fixed, no, that we cannot change much. So the centrifugal force is applied on the substance, and that force increase if the rotation per minute, that is the speed of the rotation is increased, the centrifugal force also is increased. In ultracentrifugation, this rotation rate is more than lakh. Okay. From the in the clinical centrifuge, no, this instrument where this centrifugation is done, that instrument is called centrifuge. Very simple centrifuge is clinical centrifuge. Clinical centrifuge. This is in the pathology laboratory, etc. No, in medical, that separation of cell, that blood corpuscles from the blood, it's carried out by centrifugation. Okay, next question is choose the incorrect statement for ribosomes. Here is ribosome. Option A, granular, not surrounded by any membrane. This is correct. Okay, that ribosome is without membrane. Eukaryotic ribosome 80S and prokaryotic ribosome 70S. This is also correct. S stand for, say that what I told. S stand for sedimentation coefficient. This is also correct. And so all these statements are correct. So that's why D is our answer.
Sir, the question is which one is the incorrect?
No, choose the incorrect statement for ribosome. Of
Oh,
course, here, okay, say here, this one, choose the incorrect. If it is given incorrect, then all are correct. But this one is, if we see, then see here, this one you have to, mm, this one you have to do like this. See, what is, see this one, this is, these are describing the ribosome. No, these two statement A and B, these two statements are describing the ribosome. Either this question has been written wrong, choose the correct, or this S stand for sedimentation coefficient, that is true, but this one is not describing any way ribosome. For ribosome, but for ribosome, this one also is, this is not describing the ribosome. This is describing what is S. And if all of the above are incorrect, no, all of the above are not incorrect. All of the above is not incorrect. So then this one we can see as a, this will come as an answer. This cannot be that all of the above is incorrect. No, all are correct statements. This one is, this is the wrong. Okay. Or actually this question, which one, choose the correct statement for ribosome. So these are correct, these are correct. This is also, it is explaining about the S. what is S in this 70S or 80S, then it is explaining this S. So in that case, all are correct, but all are incorrect if it is said that all of this above are incorrect. Incorrect. That is not true. These are correct statement. So ultimately, we have to give it here. This question is not properly structured. Okay, this one. So by here, all of the above incorrect. Which one is incorrect? All of the above incorrect. No. So this one we have to tick. Otherwise, usually this way the question is usually not asked. Okay, we will move to the next one. Now here is this is also one structure. This is the structure of the flagella. Okay, this is the structure of the flagella, and a cross section, and flagella is made of that protein. These are tubular protein, that tubulin is there. So this is A to D. We have to identify diagrammatic representation of the internal structure of, oh, this is centriole, not the flagella. No, this is not centriole. Cilia and flagella, this one here. I don't know uh, how do they write, print this question. That's why create a problem. These are from some of the book. See here, I will show you the, do you have your book with you now, right now? See, I will show your book here, and in that NCRT 11108. See here, your book. Say this one. Say this is the question. In the question, this is the structure now. Say section of cilia, flagella showing different part, electron micrograph, diagrammatic representation of the internal structure. Based on that question, And in the question it has been written what? In the question it has been written that this is written centriole, but it is not centriole, it is the flagella. So this our practice books also sometimes they make some mistake. We have to be very careful about that. So in this question here, of course, there is in the labeling little bit problem. This green line is the plasma membrane. The cilia flagella, they are covered by a membranous layer. So this is plasma membrane. In these, uh, there is peripheral microtubules. They are two, one pair you can see. That's why they are called doublet. Okay, and there is central portion, central microtubule. In the central microtubule, there are one pair. So nine pair at the periphery and one pair at the center. So this is called arrangement, that is 9 plus 2. This arrangement you can see 9 plus 2 array. So this flagella, they have eukaryotic structure is 9 plus 2. Prokaryote, it is different. Okay. So eukaryotic flagella are different. So here in this actually this is not centriole, this will be flagella. All right, this question you can change a little bit. So these are, what are these? A will be plasma membrane. B is B here. These are interdoublet breeze. C C is the central microtubule. D is the radial spoke. This is the central ring, and this portion is peripheral. So here the option is which one will be correct? A, interdoublet breeze cannot be. It cannot be interdoublet breeze. B here, plasma membrane. This is correct. B, central microtubule. No, this one also not correct. A, plasma membrane. B, interdoublet breeze. C, central microtubule. D, radial spoke. This one. Plasma membrane, interdoublet breeze. C, radial. No, this one is correct leveling. Okay, so answer is C. Here you can say this question number 87. Choose the incorrect pair. Again, cilium flagella. They have a 9 plus 2 morphology. This is correct. Axoneme, core of the cilium flagella. This is true. Axoneme is, there is core portion. Plasma membrane is there, and in the center, that core, central core portion is the axoneme. Basal body, central like structure, radial spoke, connect two Central microtubule. No, this one is wrong. So this is the answer. Hello, Samrat. Hello, hello, Samrat. Hello, students. Hello.
Yes, sir.
Sir.
You are there. No? Okay. Now come to this question. This is with chromosome. Again, diagram. Okay. Four chromosome has been shown here. All right. Four chromosome has been shown here, and this one is, say, this is some labeling is there. This is satellite secondary constriction. I think it is time to stop. Okay. Uh, so this is secondary constriction centromere. Here is centromere, shorter arm, longer arm. This is short arm again, long arm, and this centromere at the end. So what is the A, what is B? This is based on the morphology of the chromosome. What is called A? The based on the position of the centromere, chromosomes are metacentric. Very easy to remember. Meta means the middle, then submetacentric, which is not exactly metacentric, and then it is acrocentric, and telocentric. See this one, the centromere is at the end, one end of the chromosome. And so this one, A, is telocentric chromosome. B, centromere is very near to one end. So this one is called acrocentric. Here the centromere is not exactly in the middle, little away from the middle. This is the long arm, this is short arm. This one is known as submetacentric. C is submetacentric, and D is centromere at the middle, it is metacentric. So our option will be this one, A to D type of, and this is the secondary constriction, and here is the satellite. Okay, so today up to this question you can. All right, any question, anything will you ask? I think it is time to stop. Okay, I'll stop sharing.
Thank you,
Thank you,
sir.
sir.
Thank you. Welcome.
Thank you, sir.